Hi everyone. In this section, I want to talk to you about doing genome analysis. So we've already done genome assembly. Now we need to look at that genome and see what it does. And I'll apologize in advance. My kids are watching Finding Dory. So if you hear Finding Dory, that's what we're doing today. One of the first things that we need to do is examine the quality of our assembly. So when we get the assembly back, especially from Abyss, we get that file of the statistics of the quality of the assembly in terms of how many unitigs, how many contigs, and how many scaffolds. The tool that we can use to look at the output from spades, but also to really give us a better idea of, yeah, we have these pieces, but are these pieces good? Is a tool called QAST, Quality Assessment Tool for Genome Assemblies. We'll access QAST through Galaxy, just like we accessed spades through Galaxy. The developer of QAST is the same developer as spades, and that program is not supported on the Windows subsystem for Linux. So instead of having the Mac people do it one way and the Linux people do it the other way, we're all just going to do it the same way. But any of you who do have a Mac and might be interested in using it through the command line interface on your computer, please do let me know, and I'm happy to show you how to do that. And there are a couple different files that we'll download in order to help us do our quality assessment. That'll be a reference sequence from NCBI and a reference annotation from NCBI. And I'll show you how to do those things in a video where I show you how to do the assessment. One of the other things that we'll do is check for genome completeness. Did the sequencing effort appear to cover the majority of the genome? And the way that we do that is using a tool called BUSCO. BUSCO stands for Benchmarking Universal Single Copy Orthologs. And what it does is it has a database of all of the genes that should be present in a particular taxon. For example, because we're doing bacteria, we'll compare it to a list of genes that should be in all bacteria. If those genes are all present in our sequenced and assembled genome, then we can say with pretty strong confidence that it appears our genome is complete. So we will run BUSCO through QAST on Galaxy. And again, I'm going to show you how to do that in a separate video. However, you can also download BUSCO and run it through the command line interface. But because it's integrated with QAST through Galaxy, that's why I'm not having you download it specifically through the command line interface. I imagine there is support for the Windows subsystem for Linux, but I have personally not tested it on Windows subsystem for Linux. So we'll just err on the side of caution and run it through Galaxy instead. The next step, once we are satisfied that our genome is assembled to the best of the computer's ability and that our sequence is relatively, mostly complete, we do a process called genome annotation. What genome annotation is the idea that we are going to identify the genes and other features of the genome. So instead of it just being, well, I have 100 contigs, good luck, it's, this gene is on this contig and this RNA is on this contig. And so it allows us to identify what genes are present and where they are on our sequenced genome. Because really, what's the point of having the assembled genome if we have no idea what is found in that genome? And these processes are done for both prokaryotic and eukaryotic genomes. And remember, I'm only having us do prokaryotic genomes simply because they're smaller and they're faster. So it's easier to kind of iterate through a prokaryotic genome and get all of these processes done. But for example, Abyss and spades can both be used to do eukaryotic genomes as well. It just takes forever unless you have a really powerful computer. So again, these are not only limited to prokaryotic genomes. You can use them for eukaryotic. Some of the tools that we'll talk about in a little bit for annotation are specific to prokaryotes. However, there are eukaryotic options as well. So if you end up being interested in doing eukaryotic genomes, we can chat and I can give you some of those ideas for tools as well. There are two types of annotation that we're going to do when we do our annotation. Well, the computer will do it for us, but it's two things that are happening. One is a structural annotation. So by structural annotation, what that means is that the computer is looking for example, start and stop codons to predict an open reading frame that could be a gene. It's looking for promoter motifs, potential operons, any introns, which again, we don't have in prokaryotes for the most part, but a eukaryotic annotator is going to do that. Exons, right? Again, mostly going to be in eukaryotic systems, but that's part of the structural annotation. And all of the various RNAs, like a tRNA, rRNA, non-coding RNA, all the different kinds of RNAs. The other step of annotation that the software can do is called a functional annotation. And that's basically looking at the gene or the predicted ORF and saying, oh, this looks like Rec A, or this looks like a toxin, or this looks like this kind of protein. This RNA looks like this type of RNA. So it assigns a predicted function to all of the genes. So both of those combined are really important and give us that picture of what a genome annotation looks like. Now, what you have to keep in mind is this idea of, I think I've said this before in class, but I'm sure you've heard it other places too, garbage in, garbage out. If you put in a garbage assembly, you're going to get a garbage annotation. So that's why we do the quality steps to make sure it looks as complete as possible. We have as few scaffolds or contigs as possible, so that we get what we can rely on as a relatively decent annotation. And this also is why, for example, with that GEP, the Pathways project, or the WASP annotation project, that we go in and manually check these things, right? So we have to go in sometimes and manually check and make sure everything is good. Other things, if there's a large gap, if for whatever reason a large region of the genome didn't get sequenced or it couldn't be assembled, obviously we can't annotate that. If a sequence seems to be kind of ambiguous, maybe the sequencing quality wasn't great in a particular region, that can be difficult to annotate. And if you've discovered a new organism, the structural annotation is usually pretty easy. ORFs, right? An open reading frame looks the same pretty much regardless. You have a gene or you have a sequence of DNA. It has what looks like a ATG, lots of open space before you finally have what appears to be a stop codon, right? Things like rRNAs look the same, tRNAs look the same. So that structural annotation is usually pretty straightforward. A functional annotation, at least in genes that are highly conserved, is really easy. But if you find a new organism that potentially has new functions, it can be really challenging to annotate and describe the function of these unknown genes. And, we also like to think about the idea that, well, since we've sequenced everything from E. coli to people, we know what a lot of these genes do. But even within E. coli, there's a relatively high percentage, I want to say something like 20% of the genes in E. coli, we don't actually know what they do. So if we can't even figure out everything that E. coli does, it's going to be a challenge, especially when we're talking about eukaryotic organisms. So we're going to use two different tools for doing annotation. Again, we're using a tool that is going to be through the command line interface, and we will be using a web-based tool. So the tool that we're using through the command line interface is a specific prokaryotic genome annotator. It also can be used for viral genomes, so it's meant for small genomes. It's called PROKA, Prokka. It's apparently an abbreviation for prokaryotic annotator. It was designed for prokaryotic and viral genomes, and it enables rapid, accurate annotation. So when you start this process, it should run for less than an hour before it's finished. And that's just on a standard, like not a high performance computer, just on a standard computer. So there are options where you can customize for you have specialized needs. You can create a custom database if you're looking for something specific. Maybe you don't care about the whole genome, you're only interested in certain things. And one of the nice things about Proca is that its output is acceptable to deposit into NCBI. So when you sequence a genome for the first time, or like let's say JGI, right? JGI sequenced all those genomes that you're analyzing for your MRA paper. So they did the assembly, which we did yesterday or Tuesday, and they did the annotation, which we're doing with this set of slides. And you have to put all of those files into NCBI to get it published. You have to put in the raw sequencing reads, you have to put in your assembly, and you have to put in your annotation. So the output from Proca is acceptable for putting directly into NCBI, which makes it really nice. Now, I guess the downside would be that it uses local databases, meaning the databases are on your computer. It doesn't pull from the internet. And that's why it's faster, because it's just kind of working on your computer, you know, bing, 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 and then it's done. But that means you may need to update it periodically if new genes are identified or new proteins are characterized as the databases get updated. The other tool that we're going to be using is called RAST, Rapid Annotation Using Subsystems Technology. It's a website. The link is here in the slides. It is also an automated system for identifying protein encoding genes. It also will attempt to place it into the context of like metabolic pathways, various subsystems that are integrated. It tries to give you a more integrated and graphical representation of the contents of your genome. So RAST is really great as well, especially for exploring the content of your genome. And the combination of using Proca and RAST can give you a lot of information about a prokaryotic genome. In order to use RAST, you will need to register for an account, and that can take up to 24 hours. So I will caution you right now that it can take time for your account to be approved. So I recommend, like, as soon as you're watching this video, pause the video and register for your account. Like right now, stop the video, register for your account, and then come back to the video once you've registered for your account, because you will have an assignment that you need to do with RAST. So this is what it looks like when you're in RAST. And you see this is like a super old screenshot, but honestly, it looks exactly the same. It looks, seriously, it's been 7 years, it looks the same. There will be a tab that says your jobs. You'll just upload a new job, and that will be the config file that you'll upload into there. And I'll show you how to do all this in a separate video, like for the activity for Thursday. And then, it'll run and it'll tell you when it's done, and then you can explore all of the different options. And so it'll give you something like this, different pathways. It'll give you the size, the GC content, the N50. It'll give you all of that information.